Hey, hello everyone. I'm happy to be here to share with you a brief overview of my PhD project, and I'll be talking about sexual dimorphism in dorsal cluster neurons and fixation behavior in fruit flies. First, a brief overview of what we're doing in our lab. So the core focus of our lab is to study individuality and how it arises and its origins. And right now we're tackling this question by studying several behaviors, such as decision-making, object orientation, navigation, and sleep. And within my project, I focus mainly on fixation behavior. And this behavior is a component of the fly's innate object orientation response. And what it does is it allows the fly to detect and fixate on a visually salient or remarkable landmark and approach it. And this can be as simple as a vertical black bar or stripe. And we leverage this innate behavior in a setup called Buridan's assay that you can see here. And you can see there's a platform and surrounded by water. And instead of presenting the fly with one stripe, we present it with two equally appealing stripes and see how it behaves. And we track it for 15 minutes on this platform. And so we end up with like a wide spectrum of behaviors that range from, as you can see here, these are 8 different individuals. We have some flies like individual 1 that completely ignore the stripes, and all the way to individual 8, where the individual fixates and goes back and forth between the stripes. And this behavior is also stable across days. So if we test the same fly on several days, it will always behave the way it behaved on the first time we tested it. And of course, this behavior is a complex behavior, and it's probably governed by several neuronal pathways, but we do know of a key player in this that plays a role in this behavior, and these are the dorsal cluster neurons. Okay. And here you can see they are interneurons in the visual system. And you can see here we have a cluster of cell bodies that sends axons all the way to the contralateral hemisphere where the cells innervate either the lobula or the medulla, and they receive input from the ipsilateral hemisphere. And by that, they can integrate visual information and relay it between the two hemispheres. And yeah, and it's important to note that not all of these cells in the cluster are going to innervate both the lobula and medulla neuropils in the visual system of the fly. Some through stochastic developmental processes, some of these cells end up innervating just the lobula, and some of them extend into the medulla as well. And we're particularly interested in these medulla axons because we have shown that there's a strong correlation between the degree of asymmetry or hemispheric asymmetry in the number of these axons and fixation behavior, in such a way that the higher the asymmetry, the stronger the fixation in Buridan's assay. And yeah, and so we quantify 2 parameters, the total axon count and asymmetry, by calculating the absolute difference between the 2 hemispheres. And here you can see just a few examples of, so these DCNs are highly variable within the same individual and between individuals. And here I present you with a few examples. Here you can see this highly symmetrical pattern that is complete. We sometimes see individuals that have reduced patterns where we have these missing chunk of axons. And here you can see asymmetrical patterns. And another special thing about these DCNs, especially when we look at these axons again, is that they are sexually dimorphic. So on average, we see males with less axons and more asymmetry than in females. So that's not to say that in females, we don't see sometimes patterns similar to those in males, but on average, we find less axons in males and more asymmetry than in females. And of course, our first line of investigation into where this dimorphism stems from revolved around the sex determination pathway. And the sex determination pathway in Drosophila involves several genes, but the most important ones are the sex-lethal, transformer, double sex, and fruitless genes. And we wanted to first see if this dimorphism is happening on a cell type specific level. And so we decided to overexpress either a male specific gene or a female specific gene in the DCNs. And what we got was that when we overexpressed a male-specific gene, we got a male-like pattern in both males and females. However, when we overexpressed a female-specific gene, we got like a contradictory effect where we also got less axons, which suggests that this is not entirely a cell type specific dimorphism. And in another set of experiments, we wanted to see how the external environment affects the DCNs in both males and females. So we raised flies at 4 different temperatures, 12, 18, 25, and 29. And we looked at the DCNs again, specifically the axons, the total count, and the symmetry. And let's start by looking at the total axon count. You can see immediately a drastic drop in the total number of axons as the temperature increases. And you can see that at all temperatures, females consistently maintain a higher axon count than males. Also, we found that the highest variability was at 25 degrees, as you can see here. And to better illustrate this dimorphism, we calculated a sexual dimorphism index. And you can see that sexual dimorphism peaks at 25 degrees, which is the optimal temperature for Drosophila. And at suboptimal temperatures, it begins to decrease. And it's at 12 degrees, which is very, very low temperature. Like below 12, Drosophila tend to not survive. So there's no significant or measurable differences between males and females at this temperature. Regarding asymmetry, it was quite similar. We only find sexual dimorphism in asymmetry at 25 degrees, and at the other temperatures, there's no difference between males and females. Also, we noticed that on average, the asymmetry was around like 2 axons. At the other temperatures, even at 25, like the range is 1, 2, or 3 axon difference. And we believe that this is a true stochastic component of this circuit because it's not being affected by temperature or by the environment and genetics. And to better understand our results, we actually referred to a recently published study in which the authors looked at the olfactory receptor neurons, specifically at their synaptic connections. And they found that as the developmental temperature increases, the synapses, the amount of synapses decreases exponentially. And what they did was they proposed this first principle metabolic model to explain this scaling with temperature. And what they propose is that there are different metabolic constraints that affect the whole body growth of the Drosophila and its brain. So for example, at 18, the body grows at a slower rate than the brain. But the body decides when the entire body stops growing. So the brain continues to grow even, like it has more time to grow more connections because the body is lagging behind. And the opposite also is true at like higher temperatures, where the body develops much faster than the brain. And they make note of like that this model could apply differently to males and females, but they do not try to explain the origins or mention like a lot about sexual dimorphism. And so we want to take it a step further and propose that there could be different metabolic constraints in males and females. And our updated hypothesis is that sex-specific metabolic differences, which we can see in body mass or developmental time may influence neural growth rates and lead to more axonal projections in female DCNs. And so now we are planning, this is just an example of one of the experiments we want to try in the future, which is to manipulate the metabolism in both males and females. For example, we can increase metabolism by enhancing insulin signaling and see how it affects the DCNs, or reduce metabolism and see if we can induce a more female or male-like pattern using a metabolic pathway. Yeah, and with that, I would like to thank you for listening and thank the organizers for everyone who participated in organizing this event. Thank you.
This last point of you always have some asymmetry, right? It doesn't change with temperature or sex, right?
Yeah, more or less, yeah.
Yeah, that makes me think again about what Buridan was proposing during the discussion. So, do you think this is just a base there that has to exist for these flies to work? The fact that you don't get damage to, that was the difference between right and left. Do you think that this has any actual meaning, as we were discussing before, that they said you need some noise in the system for the function to work? Or what are your thoughts of why this doesn't change in sex almost and in temperature?
I mean, I think that this, like, to achieve perfect symmetry, you would need like a lot of quality control. So I think that if the system can function with a little bit of noise or like asymmetry and still function robustly and perfectly, then why bother to make these corrections? That's how I think about it.
Okay, so it's not that I want to keep this because I need it to work, it's just that I don't mind if it's there.
Yeah. Yeah, yeah.
Okay.
I've got a question. Thank you. Nice. I used to come from also neurogenetics, so I've got questions. My first question is, have you tried around the more conceptual questions? If you ever wondered why, how many dorsal cluster neurons are there that you count?
Yes, there's around 40. About 40 cells in each hemisphere.
In each hemisphere. Ever played around with tweaking the number of it genetically, for example? For example, you know, like genetic regulators of that dorsal cluster neuron.
I mean, not the cell number. We only looked at how it changes with temperature, and we didn't find that. We found that temperature didn't have a very significant effect on the cell count, and also we didn't find a high correlation or strong correlation between cell count and medulla axons. So it doesn't mean that if you have more cells, because also the number of cells is variable. It's not always 40. Yeah, so, and even if you have more cells, it doesn't mean you're gonna have more axons, because not all of these cells can send axons to the medulla anyways.
Absolutely, that's right. Any idea about the mechanism for it? Like the cell, is it just 7 cells being made, or are there like, oh sorry, is it just 40 cells being made immediately, or is there apoptosis?
Yeah, they have a progenitor, and then these cells, when they're in a cluster, they have through notch lateral inhibition, some of the cells express notch, and they inhibit the cells around them in the cluster from expressing notch, and the cells that express notch are confined to the lobulas. They can't send axons to the medulla. Yeah.
Trying to link a little bit with one of the questions that Sandra was asking, and what is the implication of this noise in other phenotypes? So maybe you mentioned it and I missed it at the beginning, but you say higher asymmetry equals or correlates with higher fixation. So is higher fixation better, worse? Is there an optimal level of fixation that the fly needs to have? Or I guess I'm trying to say, should I optimize for more asymmetry, less asymmetry, average, or?
Not really, because we also, like, for example, in Canton S, we see that males are more fixated than females. But for example, in some DGRP lines, both are not fixated or both are fixated. So it depends also on the line. And you always find like you will also find females, for example, in Canton S that are fixated, and some that, I mean, our theory was first that maybe it's because females have to lay eggs, so they need to explore more and not fixate on objects and stay in place. And males, they have to like locate a female and follow the female. And so like fixation would be beneficial to them. But this is just a theory, I know. Like, yeah.
So in the earlier talk, Ben talked about the decrease in the individuality, the bias in the individuality over aging, and in your talk you mentioned the persistence of the individuality. So I was wondering how long this individuality persists.
We test the flies on 3 days. I think that we also had experiments where we tested them after a long time. I'm not sure how long, but I think, yeah, it's stable. Like
Okay,
if a fly fixates and you test it after 10 days, it still behaves the same.
So if they are different, I mean, the level of stability over long time, what makes them different in terms of the behavior? One is more stable than the other less stable.
They're all stable. If you test a fly that explores the platform on day 1 and you test it on day 10, it's still gonna explore the platform.
Yeah. So this visual behavior, your visual behavior, you say is stable, right? But the right-left tangential, as far as I remember, decreases. The bias increases over 28 days, I think you said. And I was wondering if there's a difference in persistence between behavior. I was wondering what is the mechanism behind these different levels of stability of the individuality.
I mean, 28 days also is a very old fly, so maybe it's like senescence or, I don't know, loss of function.
Yeah, but why in one behavior but not in
the other? I mean, I'm not sure we have data to show that this behavior is stable till 28 days. Like 28 days later, I don't, yeah.
You showed this decline of amount of linkage or like, and also in terms of the amount of neurons with the temperature gradient. I was wondering if you push it basically to colder temperatures, do you expect that it would go down, or do you have any ideas where actually an optimal, so most fit brain connectedness would be, or amount of axons and connections would be?
You mean this graph, right?
Difference in the growth between brain and the body. Do you have any clues of where there's an optimum?
I think the optimal temperature is 25. Like, because at 29, like we only see just the minimum amount of axons, I think, that allows this circuit to function. And yeah, at 18, I mean, we get more axons, but I don't know how that is beneficial or deleterious. I'm not sure like at which temperature it's more beneficial for the fly. Yeah. I don't get the question there. Could you repeat the question?
No, that was the question. I mean, if you have any ideas that the brain with higher numbers of axons or better connectivity would lead to better fitness. So
I mean, the only thing I found was
optimal, so.
I did find that the total number of axons correlated with how long the fly pauses while it's exploring the platform. So more axons means it pauses more, but we don't know also why or if this is beneficial. Yeah.
Thank you again.